Hi, today we will see morphology of bacteria. So morphology of bacteria. So coming to the introduction, so microorganisms are grouped under protista, which is further divided into prokaryotes and eukaryotes. So prokaryotes are the primitive organism. So this includes bacteria, blue-green algae, whereas eukaryotes are the fungi. So algae, slime, molds, and other protozoas, they belong to the eukaryotes. So bacteria, they fall under prokaryotes. So what is the major difference between the prokaryotes and eukaryotes? So if you see the major difference, nucleus is absent in case of prokaryotes, whereas in eukaryotes they have a well-defined nucleus. And deoxyribonucleic acid is absent in prokaryotes and it is present in eukaryotes. It divides by mitotic division in prokaryotes, whereas in eukaryotes they do not divide by mitotic division. And other cell organelles like the cytoplasmic reticulum, your mitochondria, Golgi apparatus, endoplasmic reticulum, and the cytoplasm, they are all absent in prokaryotes, whereas they are present in eukaryotes. So they possess, the bacteria possess usually extra nucleic genetic elements, which is called plasmids or episomes. They are transmitted to the daughter during binary fission, and they are transferred from one bacteria to the another. So next we will see the shape of the bacteria. So shape of the bacteria, depending upon the shape, we can classify into cocci and bacilli. So cocci are nothing but they are oval or spherical cells, whereas bacilli are rod shaped. So based on the arrangements, this cocci can be arranged in groups, pairs or in chains. And bacilli, they are arranged in chains, pair, and some bacilli are curved, comma shaped or cuneiform shape. So we can see the clusters like arrangement, or it can be in chains, tetrads, octate or in pair. So they can be this bacilli. They are usually scattered, but they can be arranged in cuneiform or in chains pattern. So there are other shapes, that is spirally coiled shape of bacteria, which is seen in spirogates. So coming to the shape of the bacteria, the shape of the bacteria, already I told you, you have based on the gram staining, gram positive cocci and gram negative cocci, gram positive bacilli and gram negative bacilli. So later I will be telling what is gram positive or negative. So coming to the shape of the bacteria, based on the bacterial cell anatomy, you can the shape of the bacteria is usually rigid, has a rigid cell wall or plasma membrane, and there are also inclusions, that is lysosomes, ribosomes, and diffuse nucleoid material containing single circular chromosome. So there are some appendages like the capsule, flagella, and fimbriae. So, as already I told you, based on the gram staining, these are the examples of the bacteria. Gram positive cocci, which is arranged in clusters, is Staphylococcus. In chains, it is Streptococcus. Pairs or lanceolate shape, Pneumococcus. Pairs or in chains or spectacle shape, so it is Enterococcus. Tetrads, Micrococcus. Octate, Sarcenia. Gram-negative cocci arranged in pairs, that is a lens shape, that is Gonococci, and kidney shape is this is Meningococci. that is lens shaped is meningococci and kidney shaped is gonococci. These two are gram-negative cocci. So classification of the bacteria depending upon the morphology and gram-staining property. Gram-positive bacilli are arranged in chains. That is, it gives the bamboo stick-like appearance. That is Bacillus anthracis. Chinese letter or cuneiform pattern. Cornybacterium diphtheriae. Then palisade pattern is in diphtheroids. Branched and filamentous forms. That is actinomyces and nocardia. So classification of the bacteria depending upon the morphology. You also have this gram-negative bacteria, Haemophilus or Proteus, and thumbprint appearance is seen in Brodella pertussis, comma-shaped Vibrio cholerae, curved, that is Campylobacter, chains, Streptobacillus, spirally coiled or flexible spirochetes, rigid spiral forms, that is spirillum, and bacteria that lack cell wall is Mycoplasma. So let us see each a part of the bacteria in detail. So first we'll see the bacterial cell wall. So the bacterial cell wall is a very important structure. So it is made up of, it is usually tough and rigid and it gives the shape to the bacteria. It is around 10 to 25 nanometers in thickness and weighs 20 to 25% of the dry weight of the cell. So the bacterial cell wall function is to protect the cell against osmotic lysis. It confers rigidity, presence of peptidoglycan layer in the cell wall. It also protects from the toxic substances. There is a site of action of several antibiotics. And there are some virulence factors, endotoxins, which also contribute to the pathogenicity. So the antibody against this specific cell wall antigens, they provide immunity against the bacterial infection. So differences between the gram-positive and gram-negative cell wall. So if you see the gram-positive cell wall, it is made up of peptidoglycan layer and it is made up of a thick peptidoglycan layer, and there is also presence of teichoic acid which is present in gram-positive cell wall, and the lipid content and lipopolysaccharides are all present in the gram-negative cell wall, and it has a very thin peptidoglycan layer. And there are a variety of amino acids in case of gram-negative cell wall, whereas it is very few in gram-positive cell wall. And the aromatic amino acids are all absent in gram-positive cell wall, whereas it is present in gram-negative cell wall. So here you can see the thickness of the thick peptidoglycan which is seen in the gram-positive cell, whereas in your gram-negative there will be lipopolysaccharide which is loosely arranged, and there are also some porin channels which are seen in the gram-negative cell wall. So this is usually this cell wall is made up of your peptidoglycan, and it is alternating units of N-acetyl, glucosamine, and uramic acid. So you can see this thick peptidoglycan in gram-positive, whereas the peptidoglycan layer is very thin in gram-negative, but it has a lipopolysaccharide which is seen in red color. Okay. So already I mentioned, so it is a thicker peptidoglycan in gram-positive, and it is made up of mucopeptide alternating units of N-acetyl uramic acid and N-acetyl glucosamine, which are cross-linked via the tetrapeptide chains and pentaglycan bridges. So this gram-positive cell wall, if you see, It also it's composed of this uh, L-alanine, glutamine, lysine, and alanine, the other tetrapeptide chain amino acids. So here you can see the thick peptidoglycan layer. So there is also tecoic acid, which is a polymer of glycerol or ribitol, which are joined by the phosphate groups. The main structure of the cell wall. There are two types, that is cell wall teichoic acid and lipoteichoic acid. This is absent in gram-negative bacteria. So if you see the gram-negative cell wall, we have a very thin peptidoglycan layer, but we have the lipopolysaccharides which is present. So here you can have, it's mesodiamino acid which is present in the third position of the tetrapeptide chain, and pentaglycine bridge is absent in gram-negative cell wall. So outer layer is made up of a thin peptidoglycan layer and which serves as a protective barrier to the cell, and the outer membrane pore and channels are seen. So this lipopolysaccharides consists of three parts: liquid A or the endotoxin, core polysaccharide, then O side chain or O antigen or somatic antigen. So this periplasmic space in between there is a periplasmic space in between the inner cell membrane and the outer cell membrane. It encompasses the peptidoglycan layer. So we have gram-negative bacilli, gram-negative cocci, gram-positive bacilli, gram-positive cocci. So gram-negative bacilli, they usually are in scattered arrangement, whereas gram-negative bacilli, if you see, gram-positive bacilli, they have this uniform arrangement or this rambustic arrangement, as already mentioned. And cocci we have in clusters, pairs, chains, and gram-negative cocci are mostly in pairs. So this is a difference between the gram-positive and gram-negative cell wall, unsized. That is thicker peptidoglycan in gram-positive, thinner peptidoglycan in gram-negative. But variety of amino acids are seen in gram-negative, whereas it's absent, it's few in gram-positive. Lipids are absent in gram-positive, whereas it is present in gram-negative. And teichoic acid is present in gram-positive, whereas it's absent in gram-negative. So what is this? If you see, it is a gram-positive copy in clusters. Example will be Staphylococcus. So this picture shows gram-positive cocci in pairs. So example will be Streptococcus pneumoniae. So this is a gram-positive bacilli containing spores, spore bearing. This is gram-positive cocci arranged in chains. Example will be a Streptococcus pyogenes. This is gram-positive bacilli. So here you can see the gram-negative cocci in pairs. This is gram-negative bacilli. So how do you see the bacteria? Basically by simple staining, gram-negative staining using Indiaine, differential staining using gram stain, then impregnation technique, that is Fontana staining technique. So simple staining, what is simple staining? So simple staining is if you use a single dye to stain the bacteria, it is called as simple staining. So what is differential stain? If you are using more than two dyes to differentiate between gram positive and gram negative, in that case it is called as differential stain. So what is the principle of gram stain if you see? So usually an acidic protoplasm takes up the basic dye and it retains it. by fixing it with iodine, which we add in one of the steps of gram stain. And moreover, it has a gram positive will have a thick peptidoglycan and it will retain the primary dye, whereas your gram negative will take up the secondary dye because it has polysaccharide, that is lipopolysaccharides, which dissolves on decolorizing, and then it will your after decolorize your lipid layer is dissolved by using acetone. The diiodine complex will come out of the bacterial cell, and then you can see that it will take up the secondary dye. So that's why it is pink in color. This is the principle of gram stain. And negative staining we use to where gram stain cannot be used. In case of in demonstrate this capsule, there you can use this indaying staining. And we have impregnation technique where very thin bacilli or very thin organism we can without which cannot be seen by gram stain, you can use this impregnation technique. So next, coming to the cell membrane. The cell membrane is essential for the survival of the bacteria. So there is this fluid mosaic model of the cell membrane, which is the widely accepted model. So basically there is 5 to 10 thick bilayered phospholipid and several proteins which are embedded in they are the integral and peripheral proteins. So they lack sterols such as cholesterol except in mycoplasma. They contain pentacycline cyclic sterols like molecules which are called as haploids. So what is the function of the cell membrane? Below the cell wall we will have the cell membrane. So this is a semi-permeable membrane. It transports the nutrients and it's also a site for the metabolic processes. Coming to the cytoplasmic matrix. Cytoplasmic matrix, this bacterial cytoplasm lacks this membrane-bound organelles. So it's basically composed of water, which is 70%, and then it's packed with ribosomes, storage granules, inclusions, and cell membrane invaginations, which are the mesosomes. So the plasma membrane and which are everything within it, it's called as a protoplast. So coming to the ribosomes, they are the site for protein synthesis. They are composed of rRNA and ribosomal proteins integrated with the mRNA to form polysomes. So there are also genetic codons which are seen in the mRNA which are translated into peptide sequence. So each 70S unit it consists of 30S and 50S subunits. So intracytoplasmic inclusion. So intracytoplasmic inclusions, it's very important. So these are the sites for energy storage for the bacteria. There are two types of inclusions. So there are organic inclusion bodies and inorganic inclusion bodies. So this granules, they are the mainly reserved materials. Usually they are the storage materials. So one of the common example of this is volutin granules, which consist of polymetaphosphate present in Corynebacterium. The special stain is Albert's or Rhesus. So this polysaccharide granules, they stain with usually iodine, and if you see the lipid granules, they stain with Sudan black. So we can demonstrate this volutin granules in Corynebacterium bacteriae by your special stain like Albert stain. So this is an Albert stain where we can see the metachromatic granules. They are the polymetaphosphate granules. So next, coming to the mesosomes, they are the invaginations of the plasma membrane. They are the shape of the vesicles, tubules, or lamella. They are prominent in the gram-positive bacteria involved in cell respiration, cell wall formation, and chromosome replication. Coming to the nucleoid. So the bacteria do not have a true nucleus or genetic material located in which an irregularly shaped region is called as nucleoid. Possess a single haploid chromosome. They are composed of supercoiled circular structure So there are also extra chromosomal DNA which are called as plasmids. So now we have just seen about the cell wall, uh, the cytoplasmic membrane, then about the intracytoplasmic inclusion bodies and ribosomes, the cell organ, with the main cell organelles. Next, we will be seeing the cell wall appendages. So, what are these cell wall appendages? So, cell wall appendages usually composed of this capsule and slime layer, flagella, fimbria, or pili. So this capsule and slime layer. So capsule and slime layer, these are the amorphous viscid layer which is present outside the cell wall, and it is a glycocalyx. So the capsule is usually well organized and it is not easily washed off. Whereas slime layer is usually diffuse and it is loosely arranged. It is one of the virulence factor for the bacteria. So what virulence factor? Because it prevents phagocytosis. So, if you see the capsulated bacteria, we have pneumococci, meningococci, Haemophilus influenza, Klebsiella, Pseudomonas aeruginosa, Bacteroides fragilis, Bacillus anthracis, Streptococcus pyogenes. So, all these except Bacillus anthracis and Streptococcus pyogenes, they are made up of polysaccharide layer, whereas Bacillus anthracis is made up of polypeptide. So what are the functions of capsules? So as already I told you, it is a main factor for virulence. So it is it prevents the cell from dying out or desiccation. It protects the bacterium from the action of lysozyme and macrophages, and it is toxic to the cell, host cell, and induces abscess formation. Example, Bacteroides fragilis. So coming to the function of capsules, other functions are we can have certain vaccines which are highly immunogenic, like pneumococcal vaccines, meningococcal, Haemophilus influenza. So these are the highly immunogenic immunogenicity of these capsular vaccines. So biofilm formation. So this, what is this biofilm formation? The bacteria can get adherent and form a matrix like. So that this and it is capable of adhering to all the plastic tissues, plastic surfaces, or any human that is catheter lumen. In that case, they form a biofilm. So, so here what will happen is they will get adhered and they colonize to the surfaces. This is also an important factor for sepsis. So how do you demonstrate capsule? So capsule cannot be seen with the gram stain. So they can be stained with negative stainings like the India ink and nigrosin. And next is your McFardian's capsule stain is another stain where you can use see this. Then we have the serological test, that is Quillen test, Quillen reaction and capsular antigen. So what is this Quillen reaction? So Quillen reaction is when we take a colony of your capsulated strain and we add some anticapsular serum, you will be having the swelling of the capsules. Then we can also demonstrate the capsular antigen. So this is a India ink staining where you can see the capsule. So flagella. Coming to the flagella, it is a thread-like appendage which protrudes from the cell wall and it confers motility to the bacteria. So it is around 5 to 20 micron in length and 0 to 0.1, 0.1 to 2.022 micron meter in thickness. So if you see the structure of flagella, we have the basal body filament, basal body, and hook. So this is a filament which protrudes outside, and the curved part is a hook, and this is a basal body to which it is attached. So based on this arrangement we have, so basically this is a organ of locomotion. So based on the arrangement we have monotrichous, locotrichous, peritrichous and amphitrichous flagella. So monotrichous flagella is, we have this vibrio, um, where only at one end we can see the flagella. So lophotrichous flagella is spirillum, peritrichous is Salmonella typhi or E. coli, amphitrichous is Alcaligens faecalis. So peri is all around, lopho is one end, amphi is at both the poles. So how can we detect this flagella? So flagella can be detected by demonstrated with the tannic acid staining or with the electron microscopy. So the indirect means of demonstrating motility is by the Craig's tube method, where we keep a tube within the test tube having the motile strain. We can see the organism motile. Uh, we just keep the agar outside under tube. Okay, in the tube we put the motile strain. Then if we incubate, we can see the motile strain growing outside. That is because of movement. So that can be demonstrated by the Craig's tube method. Then hanging drop, where we keep a drop over a cover slip and see the cavity slide. Where when the drop hangs, the cavity slide, you can see the motility. Then semi-solid mannitol motility medium, where we stab the culture and we look for the turbidity of the medium if it is motile. And dark brown microscopy, where we can demonstrate the motility of the bacteria. So what are the types of motility we can see? So there are different types of motility: tumbling, which is seen in listeria; gliding motility in mycoplasma; stately motility in clostridium; and darting motility. Tumbling is roll over, gliding is slowly it moves. Stately is also a static movement. Darting motility is very fast motility, which we can demonstrate in the hanging drop in case of the Vibrio cholera. Swarming on the agar plate, which is usually seen in Proteus. Then corkscrew type of motility, it is seen in spirochete. So this is about the flagella. Okay. Now we'll be seeing fimbriae or pili. So fimbriae or pili, they are the short hair-like appendages. So basically, they help in bacterial adhesion. So they are made up of a protein which is called as pilin. They are also highly antigenic. So this flagellar antigen is also antigenic. That is, they can induce antibodies, and this fimbrial antigen is also antigenic, but it is not related to motility. So basically, there are two types of pili. One is common pili, and another is sex pili. So how can you detect this fimbria? One is by electron microscopy, other is by the formation of surface pellicle formation, which is usually seen on the surface of the liquid cultures. So in case of Pseudomonas, we can detect that. Um, so, so there are atypical forms of bacteria, that is swollen and aberrant forms of bacteria, which is seen in aging cultures, that is in case of high salt concentration, and pleomorphic bacteria which exhibit great variation in the shape and size, like Proteus and Haemophilus. So there are also cell wall deficient bacteria, that is L form. So it was discovered by Klenenberg, where we can see the Streptobacillus moniliformis while studying Streptobacillus moniliformis. So this L form usually plays a role in the presence of pyelonephritis and other chronic infection. So that there are also unstable L forms where the bacteria lose their cell wall in the presence of penicillin, a mechanism of resistance which are shown by the bacteria against penicillin. It's maintained only in the presence of penicillin and can revert back to original morphology once this antibiotic is removed. So then we have this protoplast and spiroplast. So protoplast is a gram-positive bacteria where the cell wall is entirely removed, and spiroplast is they are when the gram-negative bacteria lose the cell wall, whose cell wall is partially removed. So the stable L forms, this mycoplasma lacks cell wall permanently, and they represent this stable L form. They say that they do not have a particular shape. Now coming to the bacterial spores. So far we saw flagella and then with the fimbriae and then the other atypical forms of bacteria. Now coming to the bacterial spores. So bacterial spores, they are highly resistant form of, that is the dormant stage of the bacteria. They are formed when there is unfavorable conditions. So coming to the structure of the spore, there is outer exosporium, outer coat, outer membrane, cortex, then inner membrane, and the DNA. So when there are favorable condition returns, they will revert back to the vegetative cell. So the sporulation, it refers to the process of formation of spore from the vegetative stage of the bacteria. So it is not a method of reproduction because bacteria do not divide during sporulation. So it's a complex process and it takes about 10 hours. And it is highly resistant to heat and disinfection. So when the favorable condition returns, they can revert back to this vegetative cell. So based on the shape and position, we have the central, terminal and subterminal spores. So it can be bulging or non-bulging. So these are the terminal, central, subterminal, bulging and non-bulging. So there are sporicidal agents which are resistant to they are usually resistant to the routine sporicidal disinfectants. And there are other methods where we use this, which is capable of killing the spores. And it's a great challenge in case of disinfection, all these spore forming bacteria. So how can you demonstrate the spores? They cannot be seen much, but you can see as a refractile bodies in gram stain. Modified Zeal-Nielsen stain can be used. Schoffer-Fulton stain and Muller stain are the special stain which can be used. So this application of spores is in case of sterilization control indicators. In that case, we can use that is Geothermophilus, Geobacillus dearothermophilus, Bacillus atrophius. So they are used in the sterilization indicators, and they are also used in bioterrorism, like your Bacillus anthracis. Thank you.